Good morning, class. Today we will see few more important concepts in innate immunity. So today I will be discussing the topic under these headings. First we will see the overview of innate immunity and the microbial pattern recognition by the innate immunity. So this is the important topic for today. And phagocytosis, inflammatory responses, we have already covered. Again, we will see in pictorial representation and antiviral responses and innate instruction of adaptive immune response. So this is also one more important concept. The overview of innate immunity, it is the first line of defense against microbes. So whenever microbes or any microbial organism enters into the body, the innate immunity is the first line of defense. That means it comes into action first of all, and it exists before exposure to the microbes. So that immunity exists in our body even before our body, if it does not exposed to the microbes, then also it is present in the body, and it is found in plants, insects and vertebrates. And there are three essential functions, that is initial response to microbes, and it eliminates damaged cells and initiates the process of tissue repair, and it stimulates adaptive immune response. Okay, so the three essential functions of innate immunity are: it is the initial response to microbes entering into our body, and it eliminates the damaged cells and initiates the process of tissue repair, and it stimulates adaptive immune response. Okay, so innate immunity, after entering of microbes, after some time it stimulates the adaptive response also. So that is one of the function of innate immunity. And two major type of responses seen in the innate immunity are inflammation and antiviral defense mechanisms. This slide is showing differences between adaptive and innate immunity. So if we see the response time between the adaptive and innate immunity is very much fast. That is, it takes place, or it response is within minutes or hours, whereas the adaptive immunity takes days to react. And the specificity of innate immunity, it is specific for molecules and molecular patterns associated with pathogens and molecules produced by dead or damaged cells. And whereas adaptive or acquired immunity is highly specific, it discriminates between even minor differences in molecular structure of microbial or non-microbial molecules. And diversity, a limited number of conserved germline encoded receptors are present in innate immunity, whereas adaptive is highly diverse. A very large number of receptors arising from genetic recombination of receptor genes are present in each individual. Memory responses are seen only in some of the innate immunity responses observed in invertebrate innate responses and mouse or human NK cells. And here in adaptive, there is persistent memory with faster response of greater magnitude on subsequent exposure. And self or non-self discrimination is very perfect in and no microbe specific self or non-self patterns in host. And in adaptive, it is very good. Occasional failures of discrimination result in autoimmune disease. So what is autoimmune disease? When the self or non-self discrimination uh, that discrimination is failed, that time the antibodies are produced to the self antigens. So that is autoimmune disease. So that we'll be seeing in the next coming classes. And many soluble components of blood are many antimicrobial peptides, proteins, and other mediators in case of innate. And in adaptive, there are antibodies and cytokines. And major cell types in innate are phagocytes, natural killer cells, other leukocytes, epithelial and endothelial cells. Whereas in adaptive, T cells, B cells, and antigen presenting cells are the major cell types. Okay, the response time, specificity of the response, and diversity of the response, and memory responses, self or non-self discrimination, soluble components of blood, and major cell types. So under these headings we have seen the differences between innate and adaptive immunity. So write down in your notebook this slide, it is very much important. Next, coming to the components of innate immunity. So these things I have covered in the previous class. So in just pictorial representation, again I am telling. So first line is anatomical barrier. So the anatomical barrier is that means the skin, physical barrier skin, and also chemical barriers by means of the acidic pH and antimicrobial proteins and peptides. So these will form the physical and chemical barriers to the infection. The physical barriers are epithelial layers of skin and mucosal or glandular tissues. And next, coming to the cellular response to the infection or cellular barriers, like we can see the phagocytic cells, the macrophages, dendritic cells, and then killing by NK cells, natural killer cells. So activation of adaptive immune responses after the phagocytosis, that is production of antibodies and T cell responses. And also cell activation will produce cytokines and chemokines, but that will further help in the production of fever and inflammation and antimicrobial substances leading to pathogen elimination. So these are the cellular responses. And the third one, these production of these chemicals, that is soluble proteins, complement, and then cytokines and chemokines, antimicrobial substances. These will come under the soluble proteins. So the first one is physical and chemical barrier, and the second one is the cellular response to the infection, and the third response will be the production of soluble proteins like cytokines and chemokines, antimicrobial substances, and systemic effect like fever and inflammation. These two are the further effects of cytokines and chemokines. And also complement is one more important protein in the innate immunity. So complement system of innate immunity that I will be telling in the tomorrow's class. So now coming to the microbial pattern recognition by innate immunity. So I have told you this is the main topic for today. So the cellular responses of innate immunity, the immune system recognizes two types of patterns associated with the pathogens. One is PAMP and other is DAMP. PAMP means pathogen associated molecular pattern. That means every pathogen, microbial pathogen, is having a molecular structure which is required for their survival, pathogen survival. So that molecular structure of the microbial pathogen required for their survival is the PAMP, pathogen associated molecular pattern, and DAMP is damage associated molecular pattern. So any damage, and it is the result of cell damage by infections or stress or any transformation or mutation. So that cell, if it is damaged by these types of infections or stress, that is recognized by the immune cells. So it is called damage associated molecular pattern. And the cell receptors are called pattern recognition receptors. So these patterns are recognized by specific receptors on the cells. So that is why it is simply called as pattern recognition receptors. So what are these? We will see in the coming slides. So PAMPs or pathogen associated molecular patterns, these are produced only by microbes and not by the host. As I have told you, it is related to pathogen. It is usually fundamental to the integrity, survival, or pathogenicity of the microorganisms. So I have told you that is these patterns are required for their survival, and it is shared by the entire classes of pathogens. So you can see the examples of some PAMPs. Nucleic acid, suppose this is having single-stranded RNA. The type of the microbe is virus, and double-stranded RNA is also virus. And CpG, it is both virus and bacteria. And the proteins like pillin and flagellin are present only in bacteria. And the cell wall lipids like LPS, so lipopolysaccharide, it is present in gram negative, and lipoteichoic acid gram positive. Like this, these patterns present for specific microbe type are called pathogen associated molecular patterns. And then the damage associated molecular patterns, suppose stress induced proteins HSPs, the patterns are HSPs. So HSPs are the heat shock proteins which that stress of heat that will induce the heat shock proteins, and crystals like monosodium urate and nuclear proteins like HMGP, that is high molecular high mobility group protein B1 present in humans. Okay, so these are the damage associated molecular patterns, like when the cell is damaged by stress or like heat shock proteins and crystals, monosodium urate and nuclear proteins, high mobility group protein B1. So in this picture you can again see the differences between innate and adaptive when compared to pattern recognition receptors. So specificity for structures shared by classes of microbes like tool-like receptors. So these are different classes of microbes. So that is recognized by the tool-like receptors present on the immune cells. So this is in innate immunity, and adaptive for structural detail of microbial molecules, that is antigens, may recognize non-microbial antigens also. So these antibodies here, the cells are having tool-like receptors or pattern recognition receptors, whereas here in adaptive the recognition will be done by antibodies, distinct antibody molecules, and the antigens are the different microbes. And here the receptors in innate encoded in the germline, limited diversity. Okay, so these pattern recognition receptors are having limited diversity. They are not very specific for every antigen. And here it is encoded by genes produced by somatic recombination of gene segments, and it is having greater diversity. And distribution of receptors is non-clonal, identical receptors on all cells of the same lineage, and clones of lymphocytes with distinct specificities express different receptors. And discrimination of self and non-self is present, and in innate immunity, healthy host cells are not recognized or they may express molecules that prevent innate immune reactions. And here it is very specific based on elimination or inactivation of self-reactive lymphocytes, and it may be imperfect also, giving rise to autoimmunity. So these pattern recognition receptors are the several families of proteins. So they come under different types of proteins. So you all know the structure of proteins and the classification of proteins. And location, these are located on the effector cells, especially importantly on the professional antigen presenting cells like macrophages, dendritic cells and B cells. So dendritic cells, macrophages, you know the tissue monocytes are the macrophages, and dendritic cells are the cells that reside in lymphoid organs and peripheral tissues. So these arise from the bone marrow and from the branch from the cells of myeloid lineage and migrate via the blood to tissues throughout the body and to the peripheral lymphoid organs. So these cells ingest and break down microbes, but unlike macrophages and neutrophils, their primary role in immunity is the frontline large-scale direct killing of microbes. So that is the function of dendritic cells and B cells. So this pattern recognition receptors are two types, transmembrane and cytosolic. So transmembrane means they are present throughout the membrane of the cell, and cytosolic is inside the cell cytoplasm. And these are of categories of three types: secreted PRRs, endocytic PRRs, and signaling PRRs. And the coming to main functions of PRRs, it is opsonization. Opsonization is nothing but making the pathogen tastier for the cell to be engulfed, and activation of complement system. So complement system is nothing but it contains different complement proteins C1 to C11, and that activation is uh, of again classical complement pathway, alternate pathway, and Manus binding lectin pathway. So these three types of complement system I will explain in detail in the next class. And phagocytosis and activation of pro-inflammatory signaling pathways. So before actual inflammation takes place, there is a signaling pathway for every cell. For inflammatory signs, what are the inflammatory signs? Fever, redness, pallor, calor, or that is heat production, and dolor. So these are the signs of inflammation. So for the production of those inflammatory signs, before some signaling pathway has to be taking place, and induction of apoptosis, that is programmed cell death. Secreted PRRs are antimicrobial peptides, alpha and beta defensins, cathelicidin, collectins, mannose binding lectin, and pentraxin C-reactive protein. So what does these pattern recognition receptors recognize on the pathogen? So they recognize the microbial membrane which is negatively charged, and finally they will kill the microbe by opsonization, microbial cell lysis, and immune cell chemoattractant. And mannose binding lectin will recognize the microbial mannan. Mannan is a protein, and it will cause again opsonization, complement activation, and cell lysis. And C-reactive protein will specifically recognize the phospholipids, bacterial phospholipids, and finally they will also kill by opsonization and complement activation and cell lysis. Antimicrobial peptides are defensins and cathelicidin. You can see here in the picture. So these are the antimicrobial peptides. So they will cause hydrophobic interactions which are very weak, and whereas they will produce the electrostatic and hydrophobic interactions which are very strong. That is the bacterial cytoplasmic membrane, which will cause opsonization by the uh, cells of the host. So finally that will lead to cell lysis. You can see here the cell membrane of the pathogen is being disrupted, and also all these cells, antigen presenting cells, will be attracted by the antimicrobial peptides released. And the mannose binding lectin, this will come under collectin group. So here this is the picture of the mannose binding lectin. You can see here MBP. It is having a carbohydrate binding site, and here one more, this C1q is a complement protein. It is having the IgG and IgM binding sites. So you can see here in this picture all the MASPs are coming and binding, and then on the microbial surface these are bound, and then it will cause cell lysis or anything, complement system activation, any mode of killing of the cell will takes place. So here in this picture it is mannose binding lectin having 2 to 6 clusters of carbohydrate recognition domains, and that will cause mannose binding lectin binds with high affinity to mannose and fructose residues with correct spacing, and here different spacing and are not bound by MBL. So some cells will not which are not specific, they does not bound to MBL, and some cells which are specific they bound to MBL. So all these pictures, these are not present in your regular textbooks, Guyton, Genong, and like a changeable. So don't worry about this by seeing these pictures. So these are all taken from Janeway's immunobiology. So you can see here Janeway and Abbas innate immunity textbook, and then one more book is QB immunology. Like that, I have searched many books, and because these are present in your competencies, PAMP, DAMP, and pattern recognition receptors. I have taken notes from all the different textbooks and made a presentation. So if you are having any doubt, you can search in the internet Janeway's immunobiology or any names of these books, and you can have these pictures. So coming to the C-reactive protein, which come under pentraxin category. So this blue and white colored one is the C-reactive protein. So it is having two phases, B phase and A phase. B facing the bacteria and A facing the cell. So that will cause the phagocytosis. These are the toll-like pattern recognition receptors you can see here. And then here C1q is the complement protein. So again it is here, it is reverse complement activation. B phase will face the cell and A phase will face the complement. And next type we have seen till now secreted PRRs. Next are the endocytic PRRs. So in endocytic we are having mannose receptors. These are C-type lectin receptors which are recognized by carbohydrates like mannose, which are present on bacteria, fungi, and parasite. And these receptors are expressed on macrophage. The cell of the body is macrophage, and these will recognize the carbohydrates like mannose which are present on bacteria, fungi, and parasite. And the final killing is by phagocytosis and NFKB, that is NK cell activation. So you can see in this picture macrophages express receptor for many microbial. So one of the receptor is mannose receptor. So other receptors are also present all over the macrophage. And then scavenger receptors recognize lipoproteins on gram-positive and gram-negative bacteria. These are the scavenger receptors. Eliminates old and apoptotic cells. Also, these eliminated old and apoptotic cells also are having, or they have to be digested by the macrophages, so that is also recognized by the scavenger receptors. And again it is by phagocytosis and NFKB activation. So after the endocytic PRRs, we will be seeing the signaling PRRs. So there are four major classes of signaling. So these are very important. Toll-like receptors which are transmembrane, and C-type lectin receptors again transmembrane, and NOD. So what is NOD? It is nucleotide domain-like receptors. This is cytosolic and retinoid acid inducible gene RIG like receptors. These are again cytosolic. So two transmembrane, one is toll-like receptor and C-type lectin receptor, and two cytosolic, that is NOD, nod-like receptors NLR and RLR, that is retinoid acid inducible gene. So these are cytosolic and these are transmembrane. So this signaling PRRs, signaling means they will cause signal of pro-inflammatory signaling pathways. This is very important slide. You note down this. Four types of signal in PRRs: toll-like, C-lectin type, nod-like, and retinoid acid inducible gene, that is RIG-like. So coming to the first one, toll-like. So toll-like receptors, they are first recognized in Drosophila fruit fly embryos in Germany in 1980, and the word toll means weird. So you can see in the picture, this is fruit fly embryo. So it is regulating the embryonic development of those fruit fly embryos, and the same toll-like protein cytoplasmic domain of toll protein is recognized, which is homologous to IL-1 in vertebrates. So they named it as toll-like receptors. So these toll-like receptors are found in large number in mammals, including human beings. So TLR4 is one of those proteins which binds to bacterial polysaccharide, and this binding initiates many intracellular events that activate transcription of genes for a variety of proteins required for innate immune responses. Okay, so these toll-like receptors are named after the first recognition of toll, weird-like thing in the fruit fly embryos. So they are having many functions, like bacterial lipopolysaccharide produced by gram-negative organism is a cause of septic shock. And TLR2, many types of toll-like receptors are present. We will see that. So it is a transmembrane structure, as I have already told you. So this is the exterior domain and this is the interior TIR domain. LLR is leucine rich repeats LRR. So ribbon model of exterior domain, and this is the TIR transmembrane. Intracellular, this is interior domain. So 13 types of TLRs are recognized. So in the first 1 to 9 they are expressed in humans and mouse, and 10 is specific to human. 11 to 13 are present in mouse. So you can see the localization of these various proteins, microbial ligands for these TLRs. So these are the microbial, endogenous and synthetic ligands. So just know that there are 13 types of toll-like receptors. 1 to 9 are human and mouse, 10 is specific for human, and 11 to 13 are present in mouse. So cellular location of TLRs, you can see the transmembrane, the exterior domain and the interior domain. The bacterial cell wall lipid, it comes and binds here in the exterior part, and this is lectin type, C type lectin receptor. This attracts the fungal polysaccharide, and in cytosolic we are having NLR and RLR. So this is again after the microbe enters into the cell, the microbial DNA and RNA. Here also TLRs are present, which will cause the here the exterior domain present here, it has entered into the inside of the microbe. So here the reverse, you can see in the previous picture also we have seen on the host cell, the part which is present exteriorly will be entering inside the bacterial cell. So in the endosome or lysosome membrane. So in phagocytosis, what I have told you, phagolysosome. Okay, after entering inside the cell, it will be entering into the inside of the bacteria. Bacteria and the cell has become fused, and that is the phagosome or endosome or lysosome. So here the exterior part will be coming into inside. Okay. So the TLR signaling pathways, after recognition of the pathogen, that will be causing signaling into the further responses to produce immune responses like inflammatory response, fever, and various killing process of the bacteria. So heat redness and heat production, all those things will be produced by these signaling pathways. Here you can see one pathway is MYD88 dependent pathway, and one more is TRIF dependent pathway mediated by TLR3 and 4. So first one, all TLRs except TLR3. So here the first one, this pathway, and the second one is here, you can see TR IF dependent pathway. So what they are finally doing, they are entering the nucleus and they are modifying the structure, nucleic acid structure. They are attaching to the nucleic acid and probably they will release some cytokines or chemokines. So TLR4 specifically they are showing MYD88 and TRIF dependent pathway. So this pathway will produce pro-inflammatory cytokines and here you can see there is production of type 1 interferons. So next coming to the nucleotide binding oligomerization domain-like receptors. So these are otherwise called node-like receptors. Nucleotide binding oligomerization domain-like receptors. These are present inside cytosolic and recognizes PAMPs and DAMPs and also stress or damage signal and similar to toll-like receptors linked to signal transduction, that is signaling pathways. These are having 23 members of NLRs. And there are three domains. If we see the structure, there are three domains: C-terminal leucine-rich repeat and central nucleotide binding and oligomerization, and N-terminal effector binding domain. So NOD is a member of CARD domain, that is caspase activation and recruitment domain. It typically associated with other CARDs containing proteins forming either dimers or trimers. So NLRC subfamily, it comes under NLRC subfamily and it is expressed on several cell types like mucosal epithelial cells, phagocytes, and responds to bacterial cell proteins like peptidoglycans. So two groups you can see, NOD1 and NOD2. So CARD is caspase activation and recruitment domain. So two types are present, NOD1 and 2. NOD1 recognizes diaminopimelic acid (DAP) derived mainly from gram-negative bacteria peptidoglycans, and NOD2 recognizes muramyl dipeptide derived both from the gram-negative and gram-positive bacterial peptidoglycans, and it is highly expressed in intestinal Paneth cells, and the mutations will lead to Crohn's disease, bacterial overgrowth leading to chronic inflammation, and gain of function leads to Blau's syndrome, systemic inflammatory disease. So homeostasis, the first slide in the picture, the left side is homeostasis and the right side is Crohn's disease. In the natural Crohn's disease, it is as a disease of gastrointestinal tract. So intestinal lumen mucus layer you can see here. This is the natural thing. Antimicrobial peptides, like when they are increased, they are taken inside the mucosal cell, intestinal epithelial cell, and they are being killed. But here in Crohn's disease, altered microbia, dysbiosis or anything taking place in the GIT because of decreased mucin and decreased antimicrobial peptides, loss of peptidoglycan signaling, loss of protective inflammatory program leading to inflammation, that is the cell bacteria or pathogens will increase and the neutrophils also will increase leading to chronic inflammatory state condition. So that is nothing but Crohn's disease. So this will be again coming in the other subjects also. So if you have any doubt, you can just Google or search in the internet Crohn's disease. So here we can see the various signaling pathways for TLR and NODs and inflammasome associated NLRs. For TLR, as I have already told you, the signaling pathways are MYD88 and TRIF, and FKB, IRF interferons are produced, and here you can see CARD and NEMO pathways are activated, So caspase activation and recruitment domain. So pro-inflammatory cytokines are released here, and here also inflammasome complex is activated by NLRs and leading to caspase 1 activation. And so again releasing the pro-inflammatory cytokines. So inflammasome is a large protein complex that activates caspase 1 to generate interleukin 1. It contains subfamily of NLRs, all these three, and non-NLR are adapter proteins like these ASC and AIM, and caspase 1 which is in inactive form. So this complex is containing all these NLR subfamily and non-NLR or adapter protein and also caspase 1 in inactive form. So finally leading to a release of IL-1 and 18, also causing the cell death of the pathogen. So variety stimuli activated inflammasome, so that is pathogenic bacteria, extracellular ATP, that will enter into the cell. So with the help of NLRP3 plus adapter plus caspase 1, all these are the inflammasome. These three are the complex. So bacterial products, crystals, potassium efflux, reactive oxygen species are affecting these inflammasome, leading to production of caspase 1 active, and this caspase 1 will help in the signaling pathway, that is conversion of pro-IL interleukin 1b to interleukin and finally secreting into the site of infection. So this is containing the monocyte, macrophage, neutrophil, dendritic cell and lymphocytes inflammasome. And next coming to C-type lectin receptors, these are the again transmembrane or plasma membrane receptors. Again all these cells are composed and recognize the carbohydrate components of fungi, bacteria, virus, parasites and some allergens. And in humans there are 15 types of CLRs are recognized which function as PRRs. So mostly they recognize sugar moieties like mannose, glucose and glucons. So they are having variety of functions like signaling and phagocytosis. You can see in this picture the C-type lectin receptors, example mannose receptor, lectin. So here in this mannose receptors and glucan receptors. So highly, mainly sugar moieties. They will recognize the sugar moieties like mannose and glucose. And CLR signaling pathways again. So you can see CLR fungal glucan. This is fungal glucan binding to CLR protein, and then CARD9, that is again CARD, caspase activation recruitment domain, and finally NFKB activation pathway and MAP kinase activation pathway, leading to production of an interferons and antimicrobial cytokine. So in the first slide we have seen that is first anatomical barrier and then the cell barriers, and then inside that is soluble proteins like cytokines, chemokines and then antimicrobial peptides. So all these tool-like receptors or pattern recognition receptors, what they will do, they will recognize the pathogen and then inside the cell they will produce these variety of signaling pathways, and finally they will lead to production of interferons and cytokine. So that is the gist of this pattern recognition receptors and PAMPs and DAMPs. So this is one defect of the protein, lectin 1 protein recognized beta glucan in the cell wall of Candida. So Candida is a fungus. So chronic mucocutaneous candidiasis, which is very common disease. So if there is defect in this protein on the host cell, so that will not be able to recognize, and this Candida fungus will produce all these lesions. Candidiasis, it is called the tongue. On the tongue you can see this patches, and also in this intestines and fingers and hands, foot, and RIG like receptors. So we have seen the CLRs, NLRs and now RLRs. So these are the soluble PRRs and they sense for various viral infections and recognize the RNA viruses in the cytoplasm of infected cells which induce inflammatory cytokines and type 1 interferons. So three members are present, that is RAG1, MDA and LGP. So these are CRD containing RNA helicase. So caspase activation recruitment domain. So till now we have seen various pattern recognition receptors and their signaling pathways. So now briefly about the phagocytosis. So the phagocytic cells are macrophages, neutrophils, dendritic cells and tissue monocytes present in the blood. So directly by PRRs recognition. So in the first step, bacteria becomes attached to membrane in vaginations called pseudopodia and bacterium is ingested forming phagosome and then phagosome fuses with lysosome and bacterium is killed and then digested by the lysosomal enzymes and the digestion products are released from the cell. So this is direct phagocytosis by PRRs. So human receptors that trigger phagocytosis, these are the CLRs and then scavenger receptors. So this is the macrophages having all these type of receptors. So in the pattern of phagocytosis by recognition, ingestion and killing. And indirect phagocytosis is by opsonization or opsonin recognition. So opsonization means to make it tasty. Recognition of soluble proteins that have bound to microbial surfaces. So soluble protein is opsonin. So opsonin receptors are collagen domain, complement receptors and immunoglobulin Fc receptors. The major opsonins are lectin, C3b and IgG. So pathogen binds directly to the phagocyte receptor and this is direct and indirect is bacteria with capsules must be coated with antibody before phagocytes can ingest them. So this coating is nothing but opsonization. Here the bacteria is being coated and then it is taken up by the macrophage. And the inflammatory response in acute phase, local inflammation takes place, tissue damage and bacteria cause resident sentinel cells to release chemoattractants and that trigger a local increase in blood flow and capillary permeability which leads to influx of fluid, exudate and the cells. So these cells, phagocytes and antibacterial substances destroyed bacteria, neutrophil and other phagocytes migrate to the site of infection. So two phases, acute and chronic. Acute is local inflammation followed by healing. Chronic is long-term, not resolved. So chronic inflammatory disease, I have told you one example, Crohn's disease. So that is long-term, which is not at all resolved, difficult to resolve. Hallmarks of the localized inflammatory response is redness, swelling, warm and pain. So this is in acute phase. Local inflammation will having these signs. Vasodilation is responsible for warmness and redness and increased permeability is responsible for swelling. That is the fluid will enter into the cell. So acute phase reactive proteins or response proteins APRs normally found in blood at low concentration and are secreted from hepatocyte of liver following stimulation by pro-inflammatory cytokines and these function as opsonins. Examples for acute phase response proteins are CRPs, mannose binding lectin, serum amyloid protein, complement factor and fibrinogen. So these are important. You remember what are the acute phase proteins means C-reactive, mannose binding lectin, serum amyloid, complement factor and fibrinogen. So local and systemic actions of the cytokines produced in inflammation. So local inflammation, systemic protective effects and systemic pathologic effect. Pathologic means after the disease has actually taken place. So TNF is tumor necrosis factor. So low cardiac output and in vessels it will cause thrombus formation and increase permeability of the vessels and in multiple tissues like skeletal muscle and insulin resistance in the pancreas. And here protective effects are in brain it will help in the production of fever of the whole body and in liver it releases acute phase protein and bone marrow it helps in the leukocyte production. So local inflammation, as we already know, the rubor, Kaler and Dollar. Antiviral response. So the major way is inducing expression of type 1 interferons, which inhibits the viral replication. The main type 1 interferons are IFN, alpha and beta, encoded by gene on the chromosome 9. These are most important in viral defense. Alpha and IFN alpha, produced from plasmacytoid dendritic cells and mononuclear phagocytes. Most potent stimuli are viral nucleic acid, and the type 1 interferon receptors are IFN, AR1 and AR2. So induction of type 1 interferons mainly by virus infected cell entering inside the cell leading to replication. All these things, microbial product PAP recognition by pattern recognition receptors, and then finally transcription factors, the signaling pathways are taking place and the interferons are released. So the released interferons, how do they act? So interferons induce expression of enzymes that block viral replication. The main thing is blocking the viral replication, so leading to finally antiviral state. So inhibition of viral protein synthesis, degradation of viral RNA, and inhibition of viral gene expression and virion assembly. So all these will lead to antiviral state. So lastly, innate instruction of adaptive immune response. So what is this prediction of adaptive response by innate immunity? So previously in insects, and so in the first slide I have told you this innate immunity is present in plants and earlier invertebrates and insects, and also in vertebrates. So this system of innate immunity is triggered by receptors that bind sequences of sugars, lipids, which are present on bacteria and other organisms, but are not found in eukaryotic cells. These sequences, so the pathogen-associated molecular patterns. So these receptors that is present on the host cells in turn activate various defense mechanisms by means of signaling I have told you. So this primitive immune system, innate immunity system, is important in vertebrates, particularly in early response to infection. Vertebrates mean in human beings and other mammals. So this is also complemented by adaptive or acquired immunity. So innate immunity is the first line of defense, but it also triggers the slower and more specific acquired or adaptive immune response. So once this adaptive response is activated, it communicates by means of cytokines and chemokines, which will further kill the virus or bacteria or any other foreign cells by secreting cytokines and activating the complement system. So stimulation of adaptive by innate. So two signal hypothesis. You can see innate immune response to microbe. So signal 1 is microbial antigen. This is the antibody. This is signal 1, and signal 2 is pattern recognition receptors recognizing the microbe. Molecule induced by innate response. Example, co-stimulated complement fragment, and lymphocyte proliferation and differentiation leading to adaptive immune response. So two signals are present in a particular cell. So both innate and adaptive. So the first line is innate, which the response takes place in minutes to hours, whereas the adaptive takes in the days period. So this is the picture again showing the pathogen having the pathogen associated molecular proteins recognized by toll-like receptors. These are the pattern recognition receptors, and then this is the T cell receptors. Here this is adaptive response. So this is expressing the receptors on the T cell and the receptors on the host cell. These are the MHC, major histocompatibility complex, and this is the antigen presenting cell. So like this, the antigen presenting cell is having both innate immune response and adaptive immune response. It can act in both the systems. So that's all for today. So I hope you understood a little bit of today's class. So though it is very difficult, so I know that this topic, this all the slides or pictures are not present in your regular textbooks. So that's why I'm suggesting you to refer the Gene-based immunobiology and Q-based immunology, those textbooks, and please see the pictures from those, or you can have this PowerPoint presentation. You can see anytime what are the PAMPs and AMPs and toll-like receptors or the pattern recognition receptors. So all these words are new to you. So these are present in the competencies. So that's why we are explaining in detail. And also this question, innate immunity system, has come for your seniors, immediate seniors in the pre-final exam. So this is very, very important topic. So immunity itself is an important concept because of recently we have that coronavirus. All the viruses, bacteria, new viruses, new bacteria are emerging, and that will be causing infections. So we should have immunity. Basically, every individual should have immunity, and we should, as doctors, we should know in detail about immunity. So that's why all these things I have explained in detail. So if you are having any doubt, you again repeat the class, listen carefully, and if you have further doubts, you can message me. So thank you for your patient listening.